Thank you, PAX Europe, for giving us this opportunity to present to you our work at Sino Biological. So today I'm going to talk about recombinant protein expressions regarding the optimization strategies and process development. So we are a one-stop reagent and service provider focused on life sciences. We have cataloged products including genes, proteins, antibodies, ELISA kits, and also CRO services from recombinant antibody and protein expression all the way to optimize process optimizations. So with that out of the way, let's go to the main topic for today, which is recombinant protein expression. So in this quick talk, I'm going to first give a very quick overview of the concept of the technology. And I would like to also show you some showcases of case studies that we do to demonstrate the optimization process that we perform here at Sino. And also recombinant protein expression is also important for virus-related research. And I'll briefly touch up on that as well. And last but not least, of course, is summary and acknowledgement. So without further ado, let's start by giving a general overview of the field of recombinant protein expression. So recombinant proteins are very important reagents in terms of life science research and pharmaceutical development. They can be used as therapeutics, and they can be added to cell cultures. It can be used for structural studies and more. And when compared to their natural counterparts, recombinant proteins are essentially have unlimited sources, and they're manufactured in a more controlled manner so that they can be very low in terms of their contaminations. For instance, they can be animal-free, and because the recombinant proteins are expressed at a overexpression system, so that the yield can be much higher comparing to natural proteins extracted from natural sources, and also the batch-to-batch variations can be controlled because everything is done in a more stringently regulated lab space. So recombinant protein expression is essentially a step up based on the central dogma, where a external gene is inserted into the host cell in the expression vector, and the host cell and then hijacks its cellular machinery to overexpress the protein, and the proteins can then be purified using chromatography methods. So this is basically the core concept of recombinant protein expression. And in terms of host cells, there have been now a great selection of host cells available, including mammalian culture cells such as the HEC293 and CHO cell lines, and also other eukaryote cell lines such as insects and yeast, and also the mostly widely used cells available is the prokaryote system, which is mainly focused on the E. coli. So different cell types have their pros and cons as listed here, and the green are the pros and the red indicates some disadvantages. At Sino, the HEC293 and CHO cell lines are our main workforce, but we also use other cell lines, mainly insects and E. coli, to take advantage of their unique features in terms of recombinant protein expressions. So the key factors that can influence the quality and quantity of the proteins are essentially every step of the way during the whole process. So if the target gene has some features, for instance, unique features, for instance, transmembrane domains, or they have highly disordered regions, or the protein have a large section of hydrophobic domains, then the protein might be considered difficult to express, or these domains could potentially affect the stability of the protein. And also the elements in the vectors and the fusion tags will also sometimes dictates the expression level as well as the stability of the protein. And of course, the transfection efficiency will determine how much vectors are inserted into the host cells, and it does determine expression level. And of course, one of the most important selection in this important key factors in this case of scenario is, of course, the selection of host cells, and which dictates the location of the protein, the post-translational modification of the protein, and the correct folding, and so on and so on, which will, of course, dictates the overall quality and quantity of the protein as well. And then, of course, the protein purification methods and protein extraction methods, as listed here, are also important. You don't want to treat the protein so harshly during the extraction, and you would like to minimize purification steps as much as possible to minimize the loss. So everything is a means to an end, but the recombinant protein expression is usually considered as a systematic work. So every step is important, and every step would need to be optimized. So main challenges that we usually observe, of course, can either be low expression levels, or proteins are formed in high levels of aggregations, or they're presented in insoluble format, which could be contributed by the protein, by some characteristics of the protein themselves, or it can be some features that are imposed by the host or the downstream processes. And of course, degradations are usually observed, and especially for transient protein expressions, we sometimes observe activity variations between batch to batch. So generally speaking, in terms of optimization strategies, there are some upstream work that we can do, which mainly focus on optimization of the vector. So at Sino here, especially for the mammalian cell culture system, we do have a unique set of optimized vectors that go with our transfection reagents and culture media. So as you can see here, so using our optimized vectors, we sometimes get much higher protein expression, protein yield comparing to other competitor brands. But that's not, of course, not all the time. But sometimes the vector does play a very important role in terms of protein yield. And then comes to the downstream processes, which includes the condition of the culture, how much time we would like to use. Sometimes longer the culture, which will result in more cell deaths, and thus releasing the cellular proteases, which will cause degradations in the target proteins. And of course, the temperature is also a factor. The temperature is also an important factor in terms of protein stability and protease releases. And and of course, transfection methods are important as well. Sometimes we would like to optimize the ratio of in this case the viral load in insect cell expression systems, so that we hit the sweet point, which is like MOI2 or 3, so we don't use a lot of viruses, but achieve the similar expression levels. And sometimes, of course, additives are also helpful. In this case, we have some anti-clumping reagents added to the cultures. So to one thing to enhance the yield of the protein. So these are, of course, very broad descriptions in terms of how we optimize protein expressions. So now I would like to show you a few case studies, demonstrate how these works are done here at Sino, and what type of strategies that we use for protein expression optimizations. So the first case study regards the expression of a tag-free homodimer protein, which is expressed in the HEC293 system. The requirements for the protein, we will have to reach milligram quality with larger than 95% purity assayed by both the gel filtration column and page. And of course, the homodimer formation is very important for the client. In this case, I would like to show you the process development that we use for the purification essentially of this protein. So because it's a tag-free, so the first step we used was an ion exchange. So the molecular weight of the monomer is at about like 20 kilodalton, and we observed bands at this region. So we collected these two fractions and moved them to a gel filtration, aka SEC purification, which removed most of the large molecular weight contaminants, and we ended up with a fraction that we consider to be mainly of our protein of interest. But of course, the smear shaped form of this band could potentially be caused by glycosylation. So at this stage, we assay the dimer and monomer ratio of the protein. We do observe that there's still a small monomer content left. Which means a second gel filtration is required. And to validate if this smear band is indeed caused by glycosylation, we deglycosylated this protein, and the smear is gone, replaced by a more sharp band, which does prove that this smeared is caused by glycosylation, which resulted in a different mobility of the protein on the gel. So, and then we took this fraction and went through a second step of gel filtration, and the final product shows a over 99% monomer content, which meets the criteria. And when assayed by a cell-based analysis, the protein is also active. So this case showed the optimizations of the purification steps is important to obtain a protein of desired quality. And the second case I would like to show is we expressed an extracellular domain of a secreted of a single pass membrane protein, which the molecular weight of the ECD is 90 kilodalton. So the host we used in this case is insect cells, and of course the protein needs to be stable and needs to have like good purity, and it needs to be secreted, which is important for the client. So in this showcase, I would like to demonstrate how the protein construct can impact the protein, the expression of the protein. So the first construct that we used, of course, we used the full ECD sequence fused with a his tag at the terminus, but we did not observe any protein secretion essentially in the culture media. So after hydrophobic analysis, we identified there's a very highly hydrophobic region at the N-terminus of the protein, which could potentially impact the stability of the protein. And based on the functional analysis, we identify this section does not interfere with the main catalytic domain of the protein. So in the second construct, we removed this highly hydrophobic region, and the protein expression and the secretion of the protein was indeed observed in the cell culture. And of course, the last and the final product is a stable protein in terms of both the page purity and in its SEC profile. All right, so the previous case indicates the protein construct is important in terms of its expression and stability. And the last showcase I would like to present here regarding the SARS-CoV-2 nucleocapsid protein, then this case demonstrated both a expression host and the impact of expression host as well as construct in terms of the protein stability and yield. So we first express this protein in the insect cells, but the protein expressed fine, but we do observe there's a substantial degree of oligomers of aggregates essentially in this prep. And even after buffer optimization, we can still observe we can still not completely eliminate these high molecular weight essentially aggregates. So we switch the host to the expression host from insect to E. coli. And in the first construct that we use, we do observe a protein expression. However, this protein is not very stable, and we observe degradations after several rounds of freeze and zone experiment. So this construct is no good. And then we move to a second construct, which we simply tweaked the tag region so that we observed a soluble expression of the nucleocapsid protein in this construct, and and during the purification process, we also introduced a small quantity of reducing agent into the purification culture, into the purification scheme, and then we observed the protein with excellent purity, and also they present as oligomers and with very good stability even after 3 times of freeze zoning. And the oligomer format of the protein is agreed very well with published records. So so the previous showcase indicates that the purification scheme, the protein construct and selection, and of course the selection of the expression host are very important in protein expression optimizations. And but again, this is as I mentioned earlier, is like a systematic process. So each step, when necessary, each step would need to be carefully tailored in order to obtain high purity and high quality proteins. So lastly, I would like to very briefly touch on the recombinant protein expression techniques for virus-related research. So for this topic, we have to mention the SARS-CoV-2. Of course, it's an RNA virus that encodes 25 proteins, which includes non-structural proteins and structural proteins. So these proteins are important for the host interaction of the ACE2 and also for diagnostic tool development as well as drug discoveries. So far, using the recombinant protein expression technique and using different hosts, especially HEC293, insects, and E. coli, we have gradually started to map the whole proteome, essentially, of the SARS-CoV-2, and we have successfully made several of its enzymes, like papain-like pronase and 3C-like pronase, as well as S protein. And we have also focused on the receptor binding domain of the S protein and created a whole library of mutants. So there are over 70 mutants available to select from in our inventory at this point. And of course, while other coronaviruses and their reagents are useful as controls, so we've also created virus proteins for the other coronaviruses as well, mainly focused on the spike protein and the nucleocapsid protein as well. And these proteins, some of these proteins are used in collaboration of the Nanomium company to create the Sinomium high throughput diagnostic chip that is also available to test any seasonal flu and also coronavirus-related upper respiratory diseases. So in summary, recombinant protein expression technique is very important to produce proteins of all shapes and forms, and optimization strategies are required to obtain high quality proteins. And this recombinant protein expression technique is also very important tools for virus related researches as well. So with that, I would like to conclude this presentation. Thank you for your time. And if you have any questions, I'd be happy to answer them either here or through email. And we do have office in Europe at Frankfurt, Germany, that can help, that can be more directly related to you. So to have our clients in both Europe and other places worldwide. Okay, thank you very much.